In this lesson, we're going to take a look at incomplete and codominance. In these cases, neither allele is acting as the dominant allele. So not all genes have dominant and recessive alleles. Some genes have alleles that are both expressed if the individual is heterozygous. And there's two different varieties of this happening. Incomplete and codominant have alleles that have three phenotypes, one for each genotype. And the genotype is expressed slightly differently for these cases because each allele is equally dominant. So we'll go through the example on the next few slides. With codominance, you will see both of the traits being shown. So you might have spotted things. Whereas incomplete dominance, you would have a blending. So a mix of red and white would make pink. So as I said, incomplete dominance is when the heterozygous individual shows a blending of the two alleles. So in this example, we have our parent generation. So one of them is big R, and one is little r. So all of the offspring are going to be big R, which means that they're all going to be pink. And this happens in snapdragons. So we'll do this as an example. So a red snapdragon is crossed with a white snapdragon. So remember, red's dominant, white is recessive. Predict the genotypic and phenotypic outcomes for the offspring. So the correct notation is actually as so. So C for color, big R for red, and then C big R. So that's one parent. And then C big W, C big W is the other parent. So the possible gametes would be, for the first parent, CR, and then for the other one would be CW, CW. I think you can probably already see what the outcome is going to be. So we'll do the test cross or the Punnett square. So we have our CR, we'll put the red at the top, CR, and the white down the side, CW, CW. So every single one is going to be CR, CW. CR, CW, CR, CW. So the genotypic frequency is that they're 100% CR, CW. And the phenotypic frequency means that they're all going to be pink. Now, when we cross some of the offspring, then you get different outcomes. But when you do a homozygous of one with a homozygous of the other, cross them together, you're going to get 100% heterozygous individuals and 100% of the blended phenotype. So we'll show this with a different example. So we have a pink snapdragon being crossed with a white one. Predict the genotypic and phenotypic outcomes of the offspring. So the parent genotypes are going to be, for the pink one, CR, CW, being crossed with a CW, CW. So the possible gametes for the first one would be CR or CW. And then the other one is going to be CW or CW. So we're going to throw that into a Punnett square. So CR, CW, and then CW, CW. So across the top line, we're going to have CR, CW, and CW, CW. And the other one's going to be CR, CW, and then CW, CW. So this time it's going to be 50% of the time they're going to be CR, CW, and 50% of the time they'll be CW, CW. And the phenotypic ratio or frequency is going to be 50% of the time they're pink, or should be pink, and then 50% of the time you should see white snapdragons. So codominance is when heterozygous individuals are showing both alleles but not blended. So if you crossed a white bull with a red cow, you would get a roan cow, which is spotted, red and white spotted. So in this example, we're being told that the red cow is being crossed with a roan bull. Predict the genotypic and phenotypic outcomes of the offspring. So the parent genotypes, the red cow is going to be CR, CR. And the roan bull is going to be CR, CW. So the possible gametes are CR or CR for the first one, being crossed with CR or CW for the other one. So we'll do our Punnett square. So we have CR, CW, and CR, CR. So we have CR, 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 CW, CR, CR, and CR, CW. So our genotypic frequency R, it will be 50% will be CR, CR, and 50% are CR, CW, which translates to 50% should be red and 50% should be roan or spotted.